In the days of the great city-states, many cities were just like fortresses, with walls to keep out all invaders. But no city could be totally sealed. Certain goods and people had to be allowed in and out. So within the walls were gates, and these were guarded.
Halt! Who comes there? Friend or foe?
If you want to get in, it pays to be recognised. And recognition is very important to a living cell. Just like a walled city, the cell has a barrier around it, the cell or plasma membrane. And this helps maintain the integrity of the cell. It keeps the contents inside and prevents the environment from flooding, from invading the cell. But just like the walled city, a cell can't be hermetically sealed. It needs to interact with the environment. For example, to take up nutrients and expel waste products. And that's where the recognition comes in. In the cell membrane are a number of tiny gates, minute channels through which specific small substances, such as nutrients or waste products, can pass in or out of the cell as appropriate. So in effect, the cell membrane is semi-permeable. Certain substances can pass across it. Now, to get a better idea of how semi-permeability operates, we need to look closely at the structure of the membrane.
There are two main types of component in cell membranes. First, there's a double layer of lipid molecules that forms a relatively flexible sheet. Their hydrophobic or water-repelling tails form an effective barrier against any water-soluble substances, either in the fluid surrounding the cell or in the cytosol. Studded within the membrane are various protein molecules. Some in the outer lipid layer, some in the inner layer, and yet others which span the membrane. It's the proteins that provide specificity in the membrane. For instance, some form the minute channels through which only specific small substances can pass.
But what if the cell needs to admit large particles, say macromolecules, much too big to pass through the minute channels in the membrane? Well, the cell can and does cope with just this situation. And it does so by methods for which our walled city has no real equivalent.
The molecular details for one such process are already beginning to emerge. In a developing chicken oocyte, the precursor of the egg cell, specific proteins can be taken up and stored in the yolk. It turns out that the cell membrane plays an important role in this uptake. First, the protein molecules on the outside of the cell bind to the cell surface. The shallow pit forming in the membrane deepens and eventually pinches off to form a spherical vesicle. This carries the molecules into the cell in a process known as endocytosis.
Once again, the structure of the membrane is vital. Just think about one aspect of endocytosis. When the vesicle pinches off inside the cell, why doesn't it leave a hole in the membrane? In other words, how does the membrane reseal? To get a better idea of what I mean, just consider a paper hoop. Leaves a hole. But what if it was liquid? Think about diving into a pool. The diver breaks the surface of the water. But unlike paper, water is fluid, so it flows back, forming a perfect seal at the surface. It may well be that the fluid nature of the membrane which it has because of its high content of lipid, is important in helping it reseal. Well, that's just one aspect of endocytosis, where the structure of the membrane is very important in ensuring that large particles get taken into the cell. But how does the cell get the right particles in and exclude all others? For that matter, how do the right particles get into the pit in the membrane in the first place? This again is a question of specificity.
And once again, specificity means proteins. In this case, proteins known as receptors. Receptors have binding sites that recognize specific substances in the cell's environment called ligands. Ligands can be hormones or growth factors, for example. There are different receptors for different ligands, and many thousands of receptors of each type. In oocytes, there are in fact special receptors for yolk proteins. In a suckling mammal, there are receptors in the cells lining the gut. These bind antibodies to the mother's milk, providing the young with passive immunity. And in other cells, receptors take up vitamins, hormones, or growth factors. The receptors cluster in pits in the membrane. In electron micrographs, there seems to be something coating the cytoplasmic face of these pits. But is this coat real or just an artifact? The answer to this came by accident. At the Medical Research Council's Laboratory of Molecular Biology in Cambridge, Barbara Pierce was studying another aspect of the cell, its internal skeleton.
I set out to isolate microtubules, that is, major structural elements of the cell cytoplasm. To do this, I had to go to the slaughterhouse and get fresh pig's brains. Unfortunately, they gave me yesterday's brains from the cold room. I took them back to the lab and ground them up and extracted them and centrifuged them. And at the end of the day, I should have had a large pellet of microtubules, but I only had a tiny pellet. But I decided to look and see what was in this pellet anyway. There wasn't a single microtubule to be seen anywhere. But what I did see were nice little structures that looked like sliced tomatoes. They were very intriguing, but I didn't know what on earth they were. But fortunately, I took a picture of them, and I showed the picture to my colleagues, and some of them recognized them as those vesicles in a basket that other people had described in nerve terminals.
It turned out that these tiny vesicles in a basket were formed by pits pinching off from the cell membrane into the cell. The vesicles were enclosed in cages less than 200 nanometers in diameter. Eventually, Pierce isolated a protein from these fragile shells. She called it clathrin, which means lattice-like. Clathrin molecules can be seen under the electron microscope. How do they assemble to form lattices? The basic building block is a three-legged structure composed of clathrin molecules. Each leg contains two protein chains plus a short tail region, rather like a boot. The trimers assemble into hexagonal lattices by changes in acidity and salt concentration. They even form closed cages, some large enough to accommodate a membrane vesicle at the center. The assembly of clathrin into cages allows the cell membrane to deform into a coated pit. If we expose a cell to antibodies against clathrin, we can see just how widespread this protein is. A fluorescent marker picks out a high concentration around the nucleus. But clathrin is also found scattered throughout the cell, lining pits in the plasma membrane. An even more detailed picture of clathrin-coated pits and vesicles has emerged using a new technique. This involves cooling the specimen rapidly to trap any water into a glass-like solid called vitreous ice. Without any crystals of ice, there's less damage to the sample. The cooling agent is liquid ethane immersed in a bath of liquid nitrogen. The specimen is clamped with forceps above the ethane. Any excess liquid is blotted off and the sample plunged into the ethane, which traps the specimen in a layer of vitreous ice. The vitrified sample can now be transferred to liquid nitrogen and mounted for the electron microscope. The micrographs are of low contrast because the Clathrin cages are unstained, but many internal features are preserved since the vitreous ice more closely resembles the normal aqueous environment of the cell. It also provides a physical support for the fragile Clathrin cages. By combining data from different tilted views of the specimen, Barbara Pierce and her colleagues were able to build up a three-dimensional model from their two-dimensional images. So what does the final model of the Clathrin coat look like? It consists of a series of nested shells enclosing the membrane vesicle. The outer red layer is the familiar cage formed by the Clathrin trimers. Below this lies a second shell formed by the terminal domains of the trimers, the boots at the end of each Clathrin leg. The innermost blue layer represents a special class of molecules called adapters. These link the Clathrin on the outermost layer with specific sets of receptors on the membrane vesicles. But how do all these components work to bring ligands into the cell during endocytosis? Remember that there are many different types of receptor on the cell membrane. To make it simpler, let's just imagine they're all one type. First, ligand binds. Many hundreds of receptors, with and without ligand, interact with Clathrin and adapter proteins to form a coated pit. The pit pinches off to form a vesicle inside the cell. But what happens next? The vesicle sheds its coat, releasing Clathrin trimers for later use. What happens now depends on the particular ligands and receptors involved. In some cases, the uncoated vesicle fuses with another structure in the cell, an endocytic organelle or endosome. Endosomes are acidic, and this brings about the release of ligands from their receptors. The ligand may be transported to another compartment of the cell for storage or breakdown. The receptors may also be broken down or returned to the cell membrane for another cycle of endocytosis.
The beauty of endocytosis is that it allows the cell to take up large particles without sacrificing specificity. Now, specificity may not be the first thing that leaps to mind when you think of a walled city. But in fact, to the residents of the city, specificity is important. After all, there's no point placing guards at your gates unless they can specifically distinguish friend from foe. So, build strong walls and place reliable sentries at the gates. But what if you're not a resident of the city? Say you're a would-be invader. Then from your point of view, strong walls and good sentries present quite a problem. You might try and sneak past the sentries, or scale the walls, or perhaps take your battering ram to the gates. But if you're an ancient Greek and good at carpentry, you might try a more subtle approach. Disguise yourself as a friendly gift and let the inhabitants of the city take you in, just like those of Troy. For a cell, specific endocytosis is undoubtedly an evolutionary success. But what if you're a virus? You can only reproduce by invading a cell. That's by getting your nucleic acid into the cell. In that case, the cell's defenses, particularly specific endocytosis, present a potential stumbling block. So, the cell's evolutionary success may mean the virus's failure. But if cells have evolved, then so too have viruses. And some viruses seem to have evolved a wooden horse strategy. They fool the cell into recognizing them as friend and use the cell's own specific endocytosis to gain entry. So, if you're a cell, beware viruses bearing gifts.
One such virus is Semliki forest virus, or SFV. It infects mice and mosquitoes and was named after the rainforest in Uganda where the virus was first isolated. SFV is an envelope virus. It contains a protein shell, or capsid, surrounded by a lipid membrane that acts as an outer covering. Within the virus is a single strand of RNA that provides the genetic information, coding for the viral proteins. Studied within the membrane of the virus are spike proteins that play a role in viral infection. But how exactly does the virus get into host cells? Mark Marsh at the Chester Beatty Laboratories in London recalls the work that began to address this question.
The viruses that we've been working with are primarily envelope viruses. These are viruses that contain a lipid membrane around their genome. The mechanism by which these viruses penetrate their host cell is through membrane fusion. That is, they must fuse their membrane with a limiting membrane of the cell. The question was, where does this fusion reaction occur? When I went to work with Ari Helenius, a colleague at the EMBL labs, he had just found that the fusion reaction for Semliki forest virus is pH dependent. That is, that it requires acid pH to trigger the fusion activity. This meant then that fusion was unlikely to occur at the cell surface. The environment around the cell is near neutral pH. And so it could potentially occur within the cell, within endocytic organelles, which were known to be acidic. The question was then, how do the viruses get from the cell surface into those acidic endocytic organelles?
They studied the entry of SFV into animal cells in culture.
Semliki forest virus is a very easy virus to use in these experiments. You can grow it up in large amounts in tissue culture. You can purify it. You can incorporate radioactive label into the different components of the virus. You can make antibodies against those components. You can see it easily in the electron microscope. You can do immunofluorescence type experiments, and it's a very manipulable virus. In addition, it's not pathogenic to man, and for that reason, it's less dangerous to handle within the laboratory.
One experiment was carried out at 0 degrees centigrade, which allows the viruses to bind to the cultured cells, but essentially slows down endocytosis. The results give a measure of how many viruses bind to the cell surface. But how many of these are actually taken up? To measure this, the experiment is repeated at a temperature that allows endocytosis to take place as normal. This can be done by incubating at 37 degrees. The entry of the virus into the cell can be followed by measuring the uptake of radioactive label. Different components of the virus can be tagged.
Metabolically, you can label the proteins of the virus with radio-labeled amino acids so that the spike protein or the capsid protein can be visualized in autoradiography. Alternatively, one can label the lipids with radioactive phosphorus or the RNA with radioactive uridine. The specific label that one incorporates into the virus using these sorts of techniques allows you to follow it. You can follow it after binding to the cell surface. You can follow its uptake into the cell, all biochemically. And one has a trace on all of the components of the virus, which allow you then to monitor how the cell copes with those different components.
Results show that the virus spends just a few minutes on the cell surface before being taken up. Under experimental conditions, several thousand viruses may be taken up per minute. How different is this uptake from normal endocytosis?
The main differences between the physiological endocytosis and the uptake of viruses is that cells normally express receptors to take up specific ligands from their extracellular media. The cells don't want to take up viruses. They're toxic to the cell and will in general kill the cell. So the cells don't express specific receptors for viruses. The viruses have to make use of receptors which are expressed on the cell surface for other purposes. The best characterized receptor to date is CD4. This is a molecule involved in the immune response, and it can function as the receptor for HIV, the virus that causes AIDS.
The route of SFV entry can also be followed by electron microscopy. Uptake into coated pits, pinching off from the cell membrane, and formation of coated vesicles. Ultimately, many viruses get taken up into endosomes. How do they release their nucleic acid into the cytosol to promote infection? In the case of SFV, the acidity of endosomes is thought to induce changes in the spike proteins of the virus. This promotes the fusion of the viral membrane and the endosome membrane, releasing the viral RNA. Is this pathway unique to SFV?
Semliki forest virus is a virus which has been worked on extensively, and the pathway is very well characterized. And from that work, it has become apparent that the pathway is used by a number of other viruses. Most notably is influenza virus, probably also viruses like yellow fever viruses. And at the moment, we're trying to see if perhaps HIV, the human immunodeficiency viruses, also use this pathway.
So for certain viruses at least, the endocytic pathway is a means of gaining entry into cells. But what other advantages does this process have for the virus?
Viruses are metabolically inert. They need the cell to replicate them. And by using the endocytic pathway, they can ensure that they are taken up into a metabolically active cell. In addition, it's becoming apparent that the viruses need to be delivered to the correct site in the cell for replication to proceed. And again, by using the endocytic pathway, they can make sure that this occurs.
Thank you.